All right, we're going to go ahead and get started here this evening. Welcome. We're going to go over the project on karyotypes this evening. But before we do that, I wanted to just talk a little bit about how this project relates to the lesson that you went over, go over some of the objectives, and then give you some of ideas about what I'll be expecting from the project. It's actually a really quick and easy project. For this genetics and heredity unit. So the lesson itself really focuses on getting you to understand the differences between DNA and RNA, describing the process of replication and explaining how mutations occur. And the project itself really wants you to focus on, putting together what you've learned in the lesson, and then being able to synthesize that information by creating a karyotype and understanding the process by which chromosomes and genes are essentially inherited. So, with that, just a couple of pieces of information. Like I said, this is really a short lesson, and the project in and of itself is short. But from a genes and inheritance standpoint, um, keep in mind that the DNA of a gene really is the blueprint for the production of enzymes of proteins. And essentially, when we're talking about genetic control, um, all of the genes that you have are really involved in controlling all of the cell activities that are present in the body. So, from a genetic influence, a particular environment really allows a gene to act and produce specific effects. Um, so with that, um, please make sure that you take a look at the chromosome theory, and also understand specifically how um, chromosomes are the focal point of what we're going to look at going forward. Okay, with a lot of these projects, the projects on Mendelian inheritance, which we'll talk about next week, and even a couple subsequents down the road, understanding, okay, essentially how or the role that chromosomes play in all of this is going to be important. Okay, so I want to point you to that specific paragraph in the lesson on chromosomes, and really for um, the points that you're going to need to understand for this project. Um, the key points are that human cells have 23 pairs um, of chromosomes. Okay, and then we also have essentially 22 pairs of autosomes, which essentially are um, the body chromosomes. Those are the information carrying chromosomes that pretty much are responsible for all the proteins in the body that are expressed when you can see. And then the final two pairs actually are what determine the sex of the individual. Okay, so in males, the last pair of chromosomes are non-identical. Okay, one is an X chromosome and one is a Y. And in females, both chromosomes are identical, and they are the X chromosomes. So those are really the high points, and that's really what's going to help you translate into this project. So let's take a quick look at this. So we're going to go over this real briefly. You've learned that genes essentially carry the inherited traits, okay, and the genes are what are found on the chromosome. So that's the blueprint that we're talking about, okay. Scientists analyze an individual's chromosomes, okay, by what using this karyotyping, okay. And a karyotype is nothing more than a map or an organized arrangement of all of the chromosomes that are found in a cell, okay. So we can fluorescent tag these chromosomes, and then magnify them to give us a picture of them. And what they actually do then is once you get a picture, you can map them using a unique banding pattern, and that's what the project is going to have you look at. Okay, the chromosomes are then paired and they're arranged by their size from largest to smallest. And if you look down here, as you move from chromosome 1 all the way down through, you can see that the chromosomes get smaller, with the exception of the sex chromosomes. Okay, so what you're going to do for this project is you're going to analyze the chromosomes, okay, using the karyotype, and it's an interactive map. And all you need to do is match these up. Okay, you need to look at the specific banding patterns and drag what you see here on the left side of the picture over to the right side and match the chromosomes up. Once you do that, okay, you'll be able to determine if this individual is a male or a female using the information that you have been able to synthesize from the lesson and looking at the map. Okay, so then essentially you have two simple questions to answer after you complete the activity. Is the individual a male or a female? And tell me how you know that based on the information that you've assimilated from the lesson. And then secondly, how many chromosomes does the individual have? Okay, you can tell me this. You can give me the number based on pairs of chromosomes or individual numbers of chromosomes. I don't care. Just please make sure that you specify that when you actually are doing that. Okay? And that's essentially the project. Okay? You're going to just tell me, you're going to complete the diagram, tell me if the individual is a male and a female, and how can you determine that, and then how many chromosomes does the individual have, both either as a pair or singly. All right. If you have any other problems, something's not working, please come into the VO, let me know. And next week we're going to get back here and go over the project on Mendelian genetics. So have a great week, everyone, and we'll see you next week.